Let's look at all the wrappings of a skeletal muscle. So we said there's a wrapping around each muscle fiber, and that's called the endomysium. And there are bundles of muscle fibers. So there is one, say this is a muscle fiber here. There's another muscle fiber here, another one here, another one here. And all these muscle fibers together form what is known as a fascicle. So you have a muscle fiber, which is actually a muscle cell, muscle fiber. They combine together to form a fascicle. So fascicle is a bunch of muscle cells. And then there are bundles of fascicles coming together. And that bundle makes the whole muscle. So when you say biceps, we are talking about a bundle of fascicles. And the whole muscle is covered by a wrapping, and that wrapping is called a epimysium. So there are three wrappings. Each muscle fiber is covered by endomysium. A bundle of muscle fibers is called a fascicle. It is covered by the perimysium. And bundles of fascicle is covered by epimysium. That's the whole muscle. And all these connective tissue fibers, finally they attach somewhere. Usually they attach to a bone, right? So they attach to a bone. So there's all these connective tissue are going to be attached either directly to a bone or through a tendon. Okay, so you might have seen a tendon, right? When you eat beef and all, you see there's a thick portion sometimes. That is the tendon, and it's very thick. It's made of thick connective tissue, and this endomysium, perimysium, and epimysium, they're all connected to this little tendon. If it is through a tendon, it can be a direct connection to the bone, or it can be through the tendon. That's how it connects to the bone, and it's a very strong connection. So we talked about this plasma membrane. The plasma membrane is called sarcolemma. L-E-M-M-A. Cytoplasm is called plasm, sarcoplasm. And inside the cell membrane, there's a lot of cylinders inside. So you have a sarcolemma, which is the membrane. Inside, muscle has got a lot of cylinders, and these cylinders are called myofibrils. It's very, very unique to the muscle. No other tissue has got these cylinders inside. So a muscle cell or a muscle fiber contains a lot of myofibrils. So this is a myofibril. And the myofibril is made of two types of protein, actin and myosin. So you're getting down, now you're coming to the real structure of the muscle. Okay. One more thing. A cell has something called a sarcoplasmic reticulum. In the muscle, the sarcoplasmic reticulum, the muscle is just called, instead of endoplasmic reticulum, it's called a sarcoplasmic reticulum. Okay, do you remember the endoplasmic reticulum we studied? Yeah, they are there's a smooth ER and there's a rough ER, all those things. In the muscle, we call it a sarcoplasmic reticulum. It's very important because that is part of the contraction process, and I'll tell you why. and how it happens. So the sarcoplasmic reticulum is the endoplasmic reticulum in a muscle fiber. And if you look here, it looks like a stocking, a mesh stocking. This is a sarcoplasmic reticulum. It's covering the myofibril. See, this is the sarcoplasmic reticulum. And there is a peculiar function for the sarcoplasmic reticulum. It contains calcium. Muscles need calcium. Calcium is a vital component for muscle contraction. So the calcium is stored in the sarcoplasmic reticulum. So the sarcoplasmic reticulum, I told you, is like a meshed stocking, but there are some enlargements in between, and these enlargements are called the terminal cisternae. So you can see this is the terminal cisternae, and these blue tubes are the terminal cisternae, and also there is another tubing in between called a T-tubule. You might have learned T-tubule in your practical class. Yeah, everybody went through that. Yeah. So there's a T-tubule. So you have the sarcoplasmic reticulum. It contains a T-tubule, and also it contains on either side it contains a terminal cisternae. So this is a terminal cisternae. This is a T-tubule. And the three together is called a triad. So this is the structure of the sarcoplasmic reticulum. And the main purpose is to provide calcium. Where is the calcium contained in? It's contained in the terminal cisternae. Okay. That's the structure of the muscle fiber. Now let's come to the sarcomere. Sarcomere is the basic contractile unit of the muscle. And a sarcomere is the area between a C disc to another C disc. So this is a sarcomere. So what happens? Inside the sarcomere, there are thin filaments and there are thick filaments. The thin filaments and the thick filaments will be sliding towards each other, and that is what we call the sliding filament theory of muscle contraction. So when a muscle is going to contract, these filaments are going to slide. Okay? And you have to imagine this is one myofibril and one sarcomere. There are thousands. Everything is going to start sliding. The length of the sarcomere is going to shorten. And when multiple sarcomeres or thousands of sarcomeres shorten together, the whole muscle shortens. And that's what we call muscle contraction. Okay? So these are the thick filaments. The thick filament is called myosin, right? And the thin filament is called actin. So you have actin and you have myosin. So let's now go into the filaments. So this is the thin filament here. This is the actin. You can see these are the actin filaments here, the red ones. But there are two other proteins there, and there is a troponin and there is a tropomyosin. So the thick filament has only myosin, but the thin filament has got actin, troponin, and tropomyosin. Actin and myosin are the main filaments. These two are regulatory filaments. So what happens is the actin attaches to the myosin head and shortening happens. So what's the role of the troponin and tropomyosin? Troponin is the key. So you have these little proteins here, the yellow, that's the troponin. So the troponin and tropomyosin they are the regulatory proteins because the tropomyosin blocks the actin from attaching to the myosin. So when you have calcium, the troponin is going to attach to the calcium. Then the troponin is going to tell the tropomyosin, move out of the way. The myosin moves out of the way. The binding sites of actin is exposed. The actin can attach to the myosin and the sliding happens. So troponin is needed, but before that, you need calcium. Calcium is, I told you, is vital. You need calcium. Calcium attaches to troponin, and that's the key. Once this attaches, tropomyosin is what blocks the attachment sites. It is now open. It can attach. The sliding filament theory comes into play, and the muscle contracts. So that's what we talked about now. We talked about the contractile proteins. We talked about the regulatory proteins. And we said calcium is absolutely essential. Okay, now coming to striations. Why is there striations in the skeletal muscle? Striation is because there are alternate dark and light areas, right? So the dark areas are called the A bands. So A band is the area around the Z line. So you can see that this contains only the thin filaments, the actin filaments. This is the A band. And this area, which is from here to here, it only contains the myosin filaments, and that's called the dark band. So you have a light band, you have a dark band. So if you look here under the microscope, light band, dark band, light band, it's alternating. So that's why we call the skeletal muscle striated. So skeletal muscle is a striated muscle. What about smooth muscle? The muscles in the viscera. Non-striated, it's smooth. Yeah. Striated muscle means it has striations. Smooth muscle means no striation. What about cardiac muscle? It is. Cardiac muscle is striated. Cardiac muscle is striated, but it's not under your voluntary control. So skeletal muscle, striated. Cardiac muscle, striated. Smooth muscle is non-striated. Okay? So the takeaway point is the A band is the dark band, I band is the dark band. I band is the light band, A band is the dark band, and I band is composed of actin filaments. A band is composed of myosin filaments. Okay, so if you want a muscle to contract, what do you need? How can a muscle contract? You need a nerve, right? A nerve has to give the muscle an order, contract. So what type of nerve do we need for that? We need a nerve which attaches to a muscle, and such a nerve is called a somatic nerve. Somatic nerve. So a somatic nerve means that nerve is under your voluntary control. Your brain, you want to move your hand, okay, you want to raise your hand or you want to lower your hand. My brain is telling the somatic neuron or somatic nerve to move the hand. That's how movement happens. So it starts from here, goes through the nerve, that nerve comes and attaches to the muscle, and that is how the muscle is given the command to move. So what happens if that nerve is damaged? You can't move. What do you call that? Paralysis. So if somebody is involved in a spinal injury, spine is where all the nerves passes through, the motor nerves. We talked about these somatic nerves, okay? That's the nerve which is passing through the spinal cord. If you have a spinal injury, sometimes you can end up with paralysis. Then what happens to the muscle? It does shrink, yeah. It not only it is paralyzed. A paralyzed muscle does not have a nerve stimulation, but a paralyzed muscle will shrink. What do you call that? Atrophy. So that is a very serious problem. So when you have paralysis of a muscle, that muscle will shrink, and that's called atrophy. What's the opposite of atrophy? Hypertrophy. That means it's increasing in size. Atrophy means shrinking in size. So atrophy, hypertrophy. So when a muscle loses its nerve supply, what do you call the nerve supply to a muscle? It's called innervation. So when a muscle is innervated, the muscle contracts and the muscle is healthy. When the muscle is losing its nerve connection, it becomes paralyzed, it is not active, and then it atrophies. Okay, it shrinks. So when the nerve is lost, you call it denervation. That is and then the muscle will shrink, and that's called the denervation atrophy. So there are two types of neurons. There's a motor neuron. Motor neuron simply means it supplies a muscle. And there's a sensory neuron. Sensory neuron means if it's cold, I feel that sensation of cold, and that is going to my brain. It's telling me you're feeling cold. So there are receptors in the skin which detects the temperature. It conveys signals to the brain, and that is called the sensory nerves. So there are motor nerves, which comes from the brain to the muscle, and there are sensory nerves from the periphery to the brain. So those are the two types of nerves. You have motor nerves, you have sensory nerves. So the direction of transmission is also different. Motor nerve starts from the brain, goes to the muscle. Sensory nerve goes from the skin to the brain. So the direction is opposite. Okay. So I introduced the word motor neurons and sensory neurons. Now I'm going to introduce one more word there. These nerves which carry the motor impulses. Motor impulses are from where to where? From the brain. So this is the brain here, and it's going to the periphery. And from the periphery to the brain is the sensory. And from the brain to the periphery is the motor. So you have motor and sensory. But these neurons are called somatic motor neurons. They supply the muscles. Somatic motor neuron. We're going to go into that in a lot of details when we learn the nerves. So I'm just setting the stage for that now. Okay? Somatic motor neurons. Okay, so in your lab, you saw a model there. And in that model, there was a nerve which was attaching to a muscle. And this is the nerve that's coming. This is a somatic motor neuron that we talked about. So that motor neuron will come like this, and it will break up into small little divisions here. And each division will end up in a little knob. And here it will be attaching to a muscle. So basically, this is the somatic motor neuron or the nerve. These are called the terminal axon. Terminal axons. And these little knobs are called the synaptic knobs. And each synaptic knob attaches to the muscle. So here you can see in this diagram, you can see the nerve coming, breaking up into terminal neurons, and then each end up in a synaptic knob. In your lab manual, it's much more detailed. So this is what we call a motor unit. So you have a nerve which is attaching to a muscle here. And let's say there is a muscle here. Let me put another color for the muscle. So this synaptic knob is attaching to a membrane here. That's a muscle here. One, there's another muscle fiber here. Two, there are three synaptic knobs, and it is attaching to three muscle fibers. Okay, and a nerve with the muscle fibers which it supplies together is called a motor unit. So you have the spinal cord here, and there's a motor nerve coming out. This is motor nerve number one. It is colored. Motor nerve number one is colored red. No, motor nerve number two is colored red. Motor nerve number one is colored blue. Blue is attached to these blue muscle fibers. Red is attached to these red muscle fibers. So all of this together is called a motor unit. So when you have damage to the nerve, all the motor units become paralyzed. And an average nerve usually has about 200 motor units or 200 muscle fibers. That's the average. But there are some nerves which only have 3 or 4 muscle fibers. Some nerves have got more than 200 nerve fibers. So what's the difference? So if you have a muscle which you want to have a lot of power, that muscle will have a lot of motor units. The number will be more than 200. But if you want to have one of the nerves, if you want to do something skillful with a lot of precision, then you'll have a smaller motor unit. So the fingers, if you're painting something, that is a smaller motor unit, 3 to 6. But when you want to lift weight, you want a huge motor unit because you need that's when you need power. Okay? So when you need power, you have bigger motor units, but when you want to have more control, you have smaller motor units. Okay, so far we were talking about the nerves coming and attaching to muscle. We didn't reach the muscle yet. We said that it's coming over here and it's going to attach to muscle. So let me show you how it attaches to the muscle now. So when a nerve attaches to a muscle, does it really go and touch the muscle? No, it doesn't, right? There is always a little gap there. So the nerve comes and it attaches, it comes here, and there's a little gap here that is called a synaptic cleft. So what is a synapsis? Synapsis is the point where the nerve meets whatever. It may be a muscle, it may be another nerve. That point of contact is called a synapsis. When it meets a muscle, we call that a neuromuscular junction, NMJ. So we are going to talk more about the neuromuscular junction because we are interested in the muscles now. So we're going to talk more about the neuromuscular junction. So you know there's an axon coming, and the axon breaks up into axon terminals, and each axon terminal ends up in a synaptic knob, and the synaptic knob comes and attaches to the muscle membrane, which is called the sarcolemma, at some special areas. It cannot just go and attach to a muscle. It has to attach to a specialized area of the muscle. And this area is called a motor end plate. It's specialized because it contains some receptors. Okay, so you can see over here, there are some purple little receptors here. Those are the areas which can receive the nerve impulses. So we are going to talk about how a nerve fiber is going to stimulate a muscle. So it happens in the neuromuscular junction. There's a nerve coming here. It is attaching to the specialized membrane of the muscle. And this membrane is called the motor end plate. Okay, I want you to note these words, motor end plate. It doesn't attach straight away. It doesn't go and hit the nerve or the muscle. There's a gap there, and that's called a synaptic cleft. So what happens there? When a nerve impulses reach there, in the synaptic knob, there are some bubbles. They are called synaptic vesicles. So the knob contains these vesicles, and these vesicles contains a chemical, and that chemical is called acetylcholine. So you have another word here, acetylcholine. And the short form for that is ACH, acetylcholine. So when a nerve impulse comes here, reaches here, this acetylcholine is released into the synaptic cleft. And this acetylcholine, which is released into the synaptic cleft, let me go to the previous diagram. What they do is that they attach to these receptors in the motor end plate or the membrane of the muscle. They attach to that receptors. When they attach to the receptors, certain things happen. So we'll talk about that in a second, but before we go into that, I want to let you know, so the muscle membrane has got what is known as a resting membrane potential. So we're going into something complicated. So you got the structure now. Oh. So we are going to talk about what happens in the muscle membrane. So the muscle membrane has got a resting membrane potential. So you have this is the resting membrane potential, the RMP, which means the inside of a muscle cell is always negative and it's minus 90. So the inside of the muscle is always having a voltage of minus 90, and we always talk about the inside. And the outside, if the inside is minus 90, the outside is positive 90. So this is the resting membrane potential. Now, why does this happen? Because there are some pumps here working. There's a sodium pump working, which pushes the sodium outside. So sodium is positive charge. It pushes the sodium outside. And there's another pump here working, which is called the potassium pump, and it pushes the potassium inside. So what happens? For every 3 sodium which is pushed outside, 1 potassium is pushed inside. So the outside is more positive, the inside is more negative. So actually it's both positive, but it's more positive and it's less positive inside. That's why we have a minus 90 resting membrane potential. Okay? So that's the resting membrane potential of a muscle. Have I lost you? No? Are you guys with me? It's a bit complicated. Yeah. Okay, the 90 means if you put a voltmeter on a muscle membrane, okay, and measure the voltage will be minus 90. Why? Because there's a sodium pump pushing the positive outside, and there's a potassium pump pushing the positive inside. But there's a disparity. For every 3 pushed to the outside, only 1 is pushed to the inside. That means the inside will be less positive. The outside will be more positive. That means the inside is negative. So if you put a voltmeter and we put one connection here and you put another connection here, the voltmeter will read minus 90. Okay? So when a nerve impulse reaches the muscle, some changes are going to happen to all this potential. Okay? And that is what I'm going to talk about next. So I'm going to show you a diagram here. So here, if you look at this diagram, you can see when a nerve fiber is stimulated by a nerve. When I said it's stimulated by a nerve, what's happening there? We stopped at, so we said there are synaptic vesicles. Synaptic vesicles release a chemical. What's that chemical called? Acetylcholine. Yeah, acetylcholine will attach to the, where does it attach? Receptors. Where are the receptors? on the surface of the muscle, on the surface of the muscle, okay? So there are some receptors there. So these acetylcholine will go and attach to those receptors. And then what happens is that this sodium potassium pump stops. Sodium, instead of, normally it's pushed outside, all the sodium will rush into the inside of the cell. So I told you the inside is minus 90, is always, but now it's going to go to minus 80. It's becoming more and more positive, right? Then it becomes minus 50, and then it becomes plus 10. It's increasing. All the sodium is going inside. Sodium is carrying all the positive charge. It's becoming plus 20, and then it becomes plus 30. When it reaches plus 30, we call that there's a reversal of polarity. It was minus 90, it went to positive 30. This is called depolarization. It's caused by the nerve. The nerve gave an order. And at the molecular level, this is what's happened. So we have what is known as a depolarization. That's what happens when the nerve gives a stimulus to the muscle, which means it's like a chemical thing which happened there. So you have a depolarization. When it reaches plus 30, then suddenly the sodium pump, which was stopped, the potassium pump and the sodium pump which stopped the potassium pump will reactivate. Okay, so it's now going to bring it back to normality. Yeah. Yes, that's right. Well, the potassium first the sodium pump stops, sodium goes inside. When it reaches plus 30, the potassium pump opens up. So potassium is pushed to the outside. So we are bringing the membrane back to normal, and that's called repolarization. So you have a depolarization happening up to 30, and then a repolarization happening, which brings it back to 90, negative 90. Yeah. So this quick depolarization followed by a repolarization, which is in succession, this is what we call the action potential. So action potential has happened. A reversal of polarity happened, and there's a repolarization happening. So these two things happen, and that's what's called an action potential. If you simply have a depolarization, that is not an action potential. It has to go back to repolarization. So a succession of depolarization followed by repolarization is called an action potential. So this action potential happens at one point of the muscle, and this will start to move to the next area. So this action potential goes to the next area, goes to the next area. It keeps moving along the length of the muscle. It completely covers the whole muscle. So this is what we call a wave, or it is an impulse. So a wave of excitation is called an impulse. And that's one property of the muscle. A muscle does not contract in one particular area. The whole muscle contracts. So when we say the muscle contracts, we reached a place where we said the depolarization and repolarization happened. So an action potential happened. Now, how is the action potential connected to the contraction? So we are going step by step. We start with the nerve. We reach the muscle. Okay. We talked about depolarization, repolarization, action potential. Now, how does a muscle contract? Okay, so that has to happen. Before that happens, something else has to happen now. Now, well, let's talk about that. So in this picture, this is called the excitation contraction coupling. That is actually how the muscle contracts. So the action potential over here reaches here. Acetylcholine is released. Acetylcholine causes the muscle to depolarize and repolarize. An action potential is created. This action potential causes the calcium ions. We need calcium, right? Causes the calcium ions to be released from the sarcoplasmic reticulum. So that's how this diagram here, you can see the action potential is reaching here. Calcium is released into the muscle cell. And it is the calcium which causes the muscle contraction. How? Calcium attaches to the troponin. Troponin will move the tropomyosin out of the way. And then what happens? The sites, the actin sites attaches to the myosin site, and then the sliding filament theory happens. And that's how the muscle contraction happens. Okay? So we talked about a lot of events now. Everything is connected, but we broke it down into small, small pieces. But you have to put the whole picture together now to understand the process. Okay? So let me take you through that process once again. So here, this is what we discussed initially. We said first, a nerve signal is arriving, acetylcholine is released. Okay, this is the acetylcholine, which is released into the synaptic cleft. And here you can see these are the receptors. Where are these receptors? In the membrane, and we call that membrane the sarcolemma, right? Sarcolemma. So here the acetylcholine comes and attaches to the receptors. And here we talked about the sodium-potassium pump opening and all, yeah. So there's a depolarization happening. All this is the depolarization. We said it reaches plus 30, right? And then sodium, potassium starts, the potassium pump starts, and it goes back into repolarization, right? Action potential is created. Okay, now we'll keep, and we said next is the excitation contraction coupling, which means now calcium is released, which means when the calcium is released, what happens? Actin and myosin sites are exposed, right? So the calcium goes and attaches to the troponin. The troponin will move the myosin out of the way. These sites are exposed. You can see these sites exposed. And the actin is able to now attach to the myosin. And that's how the sliding filament happens. Now for the sliding, so for the actin to attach to the myosin and move it, you need energy. Where does energy come from? ATP. So ATP is broken down into ADP and P, phosphate. And it is that phosphate which gives the energy for the sliding filament theory to work. Okay? So there's a lot of events happening there. When a nerve comes to a muscle, a lot of things happen there. Okay? So, yeah. All that happening, yeah. Yeah. Yeah, it's very, very complicated. But and once it is the ATP is broken down into ADP and phosphate, it's given, the power is given, then the job is done. Then it goes into a recovery phase because now we need another ATP to attach there and give the power. Yeah. That's a lot of energy consumption. And now we're going to talk about the energy, what happens, where does the energy come from? Even that is a big topic, but we'll break it down into, we'll simplify it so that you can understand how that works. So when we say relaxation, the nerve stimulation has stopped. Acetylcholine is not released anymore. There's no more excitation. But what happens? The acetylcholine is already there in the cleft, already released there, attached to the receptors. So some activity is going on there. So nature stops it by using another enzyme called acetylcholine esterase. So we have acetylcholine, which is the neurotransmitter, and we have an enzyme which breaks it down, acetylcholine esterase. That is the enzyme which breaks down the acetylcholine. So that is also very, very important. And when there's no more stimulation, the calcium which was released into the sarcoplasmic reticulum is pulled back into the sarcoplasmic reticulum. So the sarcoplasmic reticulum initially released all this calcium into the muscle cell, but now no more stimulation. It pulls back all the calcium back into the sarcoplasmic reticulum. So what happens to the troponin? The troponin does not have the calcium. Yeah, well, the block will, the troponin will come and block the sites of the actin. Okay, we said that the actin and myosin has to attach to each other to do the sliding filament theory, but now calcium is removed. The troponin says, hey, myosin, tropomyosin, move out of the way. Tropomyosin moves out, now it is blocked. So actin cannot attach to the myosin. Does it all make sense? It's simple, but there are too many steps involved. So when you go home, I want you to break it into multiple steps, understand each step so that you can understand the total process. Okay?
All of that's for contraction.
Hmm?
All of those steps are just for contraction.
For contraction, correct. Contraction, and then once the stimulation stops, the acetylcholine will be removed from there. So that's now in relaxation. Just one contraction followed by a relaxation. Okay. So there we are. So in relaxation, the calcium is removed. The troponin and tropomyosin block is back into position. The blocking sites are blocked, so no more sliding filament. So that's what happened. So when there is a relationship between the length of the muscle and the tension which is created. So when a muscle has an optimum resting length, that creates the maximum power for contraction. If a muscle is overly stretched, then that is not good. It does not contract with the force you need. Or if it is overly compressed, then you don't have enough strength. There is always an optimal length of the muscle for having an optimal contraction. And that optimal resting length is 2 to 2.5 micron. What is rigor mortis? When you die, what happens to your muscle? You become stiff. Why? Not because of blood. Blood is there, but it's not moving because there's no circulation. But that's not the reason. The reason is because when after death, the sarcoplasmic reticulum deteriorates, all the calcium will be released. The calcium goes into the muscle. The muscle does what when calcium is there? Contracts. So the muscle goes into contraction.
But that's after a few hours.
Yeah,
Right.
yeah. It has to deteriorate. The sarcoplasmic reticulum has to 3 to 4 hours. Yeah, yeah, it has to break down because till then there's some ATP and all available. So it'll still be okay. But after a while, it's gone. Then all the calcium will be released and the muscle goes into contraction. But it cannot relax because it is an relaxation needs. You have to pull the calcium out of it. Reabsorbed, exactly. So that is not possible. Also, there's no ATP available because the person is dead. So it cannot relax. So that's why it goes into contraction. That's why a dead body becomes stiff. Okay, because all the muscle, that's called rigor mortis. But later it will relax because all these muscles will deteriorate. Okay, and then once it starts deteriorating after several hours, it will start relaxing.
So let me ask you a question.
Mm-hmm. Yeah, yeah, good. Mm.
State.
That's how you?
State that set in.
Ah.
I thought they had to like break your bones to straighten you out.
Yeah, if you want to immediately, but you can leave it for a while. After a while, it will deteriorate.
Wow.
I think it's there's a set of hours for that. That's how the forensic people find out how many hours, when was a person killed. They look at, no, you don't have to break it. Just leave it for a little while. After a few hours, the myofibrils will deteriorate, the muscles will deteriorate, and then you can move it. If you want to move it immediately, yes, you have to break the bones. So let's talk a little bit about the type of exercises and all. So there are 2 types of contraction which a muscle can do. One is called the isometric contraction. If you look at this lady here, you can see that she is lifting a weight, but it's not moving. Okay, this is called the isometric. Iso means same, metric means length. So same length, the lady is taking something, holding it. So there is tension developed in the muscle, but there's no active movement. So that's called the isometric contraction. And there's another contraction called isotonic contraction, which is when there's a change in length. So this is isotonic contraction. There's a change in the length. And when you are shortening, this is shortening, it is called a concentric contraction. And the same thing can happen when you're lengthening, when you're lowering something. You could be lifting something or you could be lowering something. When you're lowering something, it's called a eccentric, isotonic contraction. And when you're lifting a weight, it is called a concentric isotonic muscle contraction. This has got implications, especially for exercise. Okay, so for any muscle contraction to happen, we need ATP, right? We need energy. And there are 2 pathways for ATP synthesis. You can have an aerobic fermentation or you can have an anaerobic fermentation, either one of them. That's how. So if you're having a short, intense exercise, say, for example, that exercise is lasting for 10 seconds, which is how much it takes for a 100-meter dash, right? So that is the time taken. The initial part of the energy will be supplied by the oxygen which is in the muscle, and that is called normally, where is the oxygen carried in the blood? Hemoglobin. Hemoglobin carries. In the muscle, it is carried by a pigment called myoglobin. Just like hemoglobin in the muscle, it's called a myoglobin. So there's myoglobin in the muscle, and this myoglobin will carry the oxygen. So initially, the oxygen is supplied by the myoglobin, but after a while, that will be depleted. And then what happens is there's a system called phosphagen system. Phosphagen system comes into play. And the phosphagen system, what it does is that it takes a molecule of phosphate, it borrows it from somewhere, okay, and it converts ADP into ATP. So this phosphate is borrowed from somewhere, and the ADP, which is freely available, is converted into a ATP. And this ATP will be used for muscle contraction. So that's called the phosphagen system. Now, phosphagen system has got two enzymes. That there is a myokinase enzyme and there is a creatinine kinase enzyme. The myokinase enzymes breaks down ADP and converts it into AMP, which is the ADP is adenosine diphosphate, AMP is adenosine monophosphate. So ADP has got two phosphate, AMP has got one phosphate, monophosphate. So you have an extra phosphate which will be used to convert ADP. This is the ultimate aim. You want to convert all the ADP into ATP, right? And it comes from that enzyme. Another enzyme which it uses is called creatinine kinase, which breaks down creatinine phosphate into creatinine, and phosphate is released. What you need is the phosphate. So initially, you have the oxygen, which is from the myoglobin, and then you have the phosphagen system. After that, what happens? After that, there's no way the muscle can continue. It has to go into the anaerobic respiration. So this is around, say, 30 seconds. And by about 40 seconds, you're well into the anaerobic fermentation, which is called the glycogen lactate system. So I want you to remember these words. First is the aerobic, then the phosphagen system comes into play, and then the glycogen lactate system, which is the anaerobic fermentation, comes into play. And after that, say after 40 seconds, what happens? Aerobic, how does that happen? Because your lungs now, then you start panting and you start producing because your lungs starts working efficiently. It takes the oxygen from the outside. It provides the necessary oxygen. So that's the sequence of events up to 40 seconds. And you can see at this stage here, after 40 seconds, what is happening? We have the lung capacity and all delivering enough oxygen, which goes back to the aerobic respiration. And aerobic respiration is what we want. When you have oxygen, you produce a lot of ATP. When you don't have oxygen, you are going into anaerobic respiration, which is very inefficient. The number of ATP produced is very minimal. Also, the fuel. Muscle has got a lot of glycogen. The glycogen breaks down into glucose, and the glucose is the fuel used for the muscle activity. What happens if your glycogen is finished? What will be the next fuel available? This is basic biochemistry. Fat, yes. After glycogen, glucose, glycogen is broken down into glucose. If glycogen is finished, next it goes into fats. It takes the fat and it breaks down the fat. But the problem with fat is that it produces a lot of lactic acid. So that's not a very efficient thing. You want glycogen, enough glycogen. But that is why where people do all these exercises and they try to burn the fat, right? That's what we just to get to the fat. But yeah, but if you keep adding a lot of glucose into the system, you won't get to the fat. Okay? That's why many diets fail. Glucose is the key, yeah. Correct. And what is muscle fatigue? When you reach a time of muscle fatigue, that means there is less glycogen, there is less ATP, there is less oxygen, and there's a buildup of all these lactic acid and all in the system. That is called muscle fatigue. So after exercise, what do? You start panting and you stop your exercise, you don't come back to normal immediately. It takes a while. Why is that? Because there's an oxygen deficit that happened. That oxygen has to be supplied to the tissue. The tissue are in a hypermetabolic state. Okay, they have already burned a lot of oxygen. There's an oxygen deficit which has happened. So the lungs has to provide excess oxygen, and it has to replenish all the oxygen which has been used, and that is called the post-exercise oxygen consumption, EPOC. That is to replenish the ATP, to replace the oxygen which was used up, to provide oxygen to the liver this is important to dispose of the lactate, and also to provide oxygen to cells which is in a hypermetabolic state. So that's why it takes a while after a heavy exercise to come back to normal. Now, if you look at the muscle fibers also, there are some differences. There are two types of muscle fibers. You have the slow twitch fibers and you have the fast twitch fibers. So the slow twitch fibers they are well adapted for endurance. They are slow, okay? And the fast twitch, they are quick, okay? And the slow twitch is the ones which you use when you're lifting weights. But if you're playing basketball or racquetball, then you're using the fast twitch muscle fibers. So Michael Jordan, which type of muscle fibers does he have? Fast twitch, yeah. Basketball is a fast game. Mitochondria, the muscle has got a lot of mitochondria because that is very, very important. But you can't increase the number of mitochondria, so we don't have it's got no relevance. But muscle is one of the tissue with a lot of, because the mitochondria are the powerhouses of the house, right, of the cell, and that's where all the ATP is produced. Two types of exercises. One is resistance training, which is lifting weights. So if you're lifting a weight, then what happens to the muscles? It increases in size. Why? Does the number of cells increase, or does the muscle fiber increase in size. Correct. That is called hypertrophy. That's the opposite of atrophy. So in hypertrophy, the muscle size is increasing, not the number of muscle fibers are increasing. So you have increase of myofibril. The number of myofibril will start increasing. That is what is called the resistance training. So lifting weight is a resistant training. Whereas if you're doing aerobic exercise, what are you improving there? Are you improving the size of the muscle? No, you're not. People who do aerobic exercise don't become bulky, but it increases the oxygen transport capacity of the blood. It enhances the cardiovascular system. So you become more efficient. You are able to have more oxygen carried to the tissue. So that is what you do in endurance training or aerobic exercise. So aerobic exercise is not for muscle building. Again, we talked about the cardiac muscle and the smooth muscle. And cardiac muscle, what is the peculiarity of cardiac muscle? Under the microscope, what do you see? You see this little intercalated disc, right? Yeah. So when you see the intercalated disc, you know that's a cardiac muscle. But I'm sorry, yeah, cardiac muscle. And the cardiac muscle is also striated. So you can confuse it with skeletal muscle, but the key is it has got intercalated disc. And cardiac muscle, can you stimulate the cardiac muscle? No, no, not voluntarily. The skeletal muscle, you can stimulate it voluntarily, but cardiac muscle is autorhythmic. It has got a pacemaker inside. It is inside the heart itself. You have a pacemaker called the SA node that will give the impulses to the heart that will cause the heart to beat. So that is smooth muscle. The big difference between a smooth muscle and a skeletal muscle is that smooth muscle has got no striation. It's not under your voluntary control, and it is what causes movement of food through the intestine. And that movement is called peristalsis. It happens without your control. You have no voluntary control over it. Okay? And I think we reached almost the end of it. Yeah. Peristalsis, yeah. Any questions? So next week we have the exam. Eh? 11, yeah. 9, I'm avoiding because you already did a worksheet. I've put a list of topics to review. Okay, so go through those topics.
That's in the lecture.
Eh? Which one?
Lecture. That like these, where you put the topics at.
Well, the topic, open your computer
Yeah, that's what I mean, like it's
in the D2L. Yeah, it's called the review questions. Yeah, just look for the, there's a, did everybody see that? Review questions? You saw it? Have a look, open your computer now, make sure you can see it, because I want you to concentrate on the review questions, okay? And so that you're kind of focused on what to study, okay?
question.
Yeah, concentrate on that, yeah. Hmm? No, you don't have to. 10 and 11, yeah. Eh? No, 8, 10, and 11, yeah. Look at the announcements, okay? That's all in the announcements. Okay, and one more thing, beyond time, make sure you bring your large Scantron, bring your pencil, okay? Today's topic was intense, right? You're welcome. Yeah,
Last time I came up to you about assignments that needed to be open.
yeah.
It was just these that,
Was
like-
it was opened, right? You
like
didn't do it?
Chapter two, that was from a long time ago, so. like,
You didn't do
I-
it? Okay. Okay, I'll open it now. I'll open, how many days do you want?
Just today,
Yes, you'll finish it today? Okay.
is
If you need anything, come to me, ask me.
a chapter assignment.
Oh, I have to stop sharing. And then, and